A Carnegie Institution for Science audio press release for September 5, 2013. Clues in Coral Bleaching Mystery. Coral reefs are tremendously important for ocean biodiversity, as well as for the economic and aesthetic value they provide to their surrounding communities. Unfortunately, they have been in great decline in recent years, much of it due to the effects of global climate change. One such effect, called bleaching, occurs when the symbiotic algae that are essential for providing nutrients to the coral either lose their identifying photosynthetic pigmentation and their ability to perform photosynthesis, or disappear entirely from the coral's tissue. Without a healthy population of these algae, the coral cannot survive. There has been much attention given to the environmental conditions that trigger a reef's demise due to bleaching, but little is certain about the precise cellular and molecular mechanisms of the bleaching process. New research from Carnegie's Arthur Grossman brings into question the prevailing theory for how bleaching occurs on a molecular level. It's published in Current Biology. Photosynthesis, the process by which plants, algae, and select bacteria convert the sun's light energy into chemical energy, takes place in a cellular organelle called the chloroplast. It's been theorized that the major cause of bleaching is the result of chloroplast damage due to heat stress, which results in the production of toxic, highly reactive oxygen molecules during photosynthesis. Grossman and his team, led by Carnegie's Dimitri Tolater and in collaboration with John Pringle and Steve Palumbi of Stanford University, demonstrated that bleaching still occurs if the algae are heat-stressed in the dark, when the photosynthetic machinery is shut off. This is surprising, since it means that toxic oxygen molecules formed in heat-damaged chloroplasts during photosynthetic reactions during the light are not likely the major culprits that cause bleaching. Therefore, other, as yet unexplored mechanisms for bleaching must exist. This work suggests the existence of potentially novel mechanisms associated with coral bleaching. A mechanistic understanding of bleaching is critical for developing strategies to mitigate or eliminate the problem of coral decline. The process of coral bleaching in the dark at elevated temperatures, and perhaps also in response to other stress conditions, could be a potential advantage to corals. Grossman explains, "One theory that we're exploring is that under heat stress conditions, the corals eject the algal symbionts at night in order to avoid the production and accumulation of photosynthetically derived toxic oxygen molecules during the day. If such molecules were to accumulate, they would threaten the viability of both the alga and its host." The team study also suggests that certain strategies proposed to protect reefs, such as shading the corals from high light, may not prevent high temperature-triggered bleaching, because bleaching would still occur in the dark. Clearly, there is a need for more thorough molecular analyses to establish the various molecular causes of the bleaching process. Additionally, Grossman and Tolater's work revealed some key details, such as the breakdown of specific parts of the photosynthetic apparatus when the corals are stressed. This process can be monitored and may potentially serve to evaluate the health of the reef and the risk of bleaching and death. With the independence for nimble pursuit, Carnegie scientists tackle some of the most profound challenges in modern science, fundamentally changing what is possible for us, our planet, and our universe.